biochemists have a unique way of solving problems in systems that are too complex for them to understand fully, right? If you go to the doctor and they take a lot of biochemistry and you have a problem, do they understand everything about the organism? Not at all, right? But do they understand enough to solve problems? And the answer is yes. And in many cases, if they're broadly extensible people, they can also solve problems that are deep in the areas where they're deep, but using the tool, the broader tool. So the broader tool emphasizes cause. So if you're driving down the street, you run over a nail, and you get a flat tire, the source of your flat tire is the nail. And the location in the street where you got it, those are all sources. But the cause is that the construction guy who was driving down the road had an open box of nails in the back end of his truck, and some of them spilled out, and there's where the cause is. It was the open container of nails, right? And when he was bouncing along, the nails were falling out, and then that was bad. So engineers at Boeing and every other engineering company in the world are constantly confronting, how deep do I go? Do I have the luxury of getting actual cause, chemistry, physics, biology, whatever the field is where the cause lies? Do I have the luxury of getting that so that I can learn from it in the future, or do I just have to find the source and replace it? And if you're not in a safety of flight environment, like you are with an airplane, a lot of the time, cause isn't gotten to. But if you work on FDA-regulated diagnostic devices, as an example, which is the field that I came from, or research devices where getting the wrong answer leads a whole group of people down the wrong path, then that's bad. And you don't want to do that, and you still want to get to the real cause. So there's a system of thought called root cause failure analysis, and it uses a tree that has normally 5 whys in it. I have a 14-year-old son, so I'm only up for 3 whys these days. But the idea is that if you have some condition B, you ask yourself, why did I get from what my problem is, how did B make that happen? And then why did B happen, and why did F happen? And pretty soon the tree grows out. And one of the most recent ones that I've worked on, not at Boeing, but at another company, had over 400 boxes in it. So it was a big tree, very, very broad. But it came back to 3 or 4 lines back here that led in to what happened. So the trick, if you're the analyst, is to make a test that's as far to the left-hand side as you possibly can so that you shut down an entire branch, like the branch that starts with B, so you don't have to do tests at every one of those blue lines that connects the boxes. If you have to do all those tests, you're going to spend a gigantic amount of money. And these things down here that are many steps deep might be seen by your boss as following a rabbit, right? So you want to solve stuff as far to the left as you possibly can. And the discipline says that you'll make something called a not list. Things that you know, K, things that you need to know, N, and those are normally the results of a test. Opinions, everybody's got one, right? And things that you think you know, and you probably already have the data for it, but you need to go and find it and bring it back, and then you can put it in the no list, or there's still a question mark in that aspect. So if you invite a whole bunch of expert people, like all of you, into a room, if you don't pay attention to their opinions, they don't want to play, right? So this is part of the human nature aspect of it. You got to allow for every opinion to at least be stated, and everyone has to be ready when this is solved and we know that G is the point of failure. Everyone has to accept the fact that A and C and D and E and H were all wrong. So people's opinions about all of those things were wrong. So if you walk into the room not ready to be wrong about almost everything you're going to do over the next 6 months, or however long it takes, you can't play the game. You got to be signing up to be wrong all the time, because you're going to be wrong most of the time, right? So if you're afraid of being wrong, you can't do it. So this is the not list. You give things numbers, you give data items, you assign people, and you assign a date when that information is going to be made available so that your boss is happy and knows when things are going to get fixed. And you can't say to your boss, we're going to be done after this set of simple tests, because you're probably going to have to verify those other things downstream. So you got to be real honest about your estimating along the way. So nucleic acid sequencing, which we all know about through the Human Genome Project, and lots of news about genetically modified organisms and the ability to detect markers for disease and so on, is a super complicated system where there's a lot of different fields all at play with many different technological components, and the way that we do it, the dirt road way, has been changed and abandoned multiple times in the history of DNA sequencing, and as a result, people have had to let go of things that didn't work or didn't work as well and grasp onto things that worked better. So some of the areas that people use to, and especially in the early days of DNA sequencing, were things like optical imaging and organic dyes and surfaces. I'm not going to cover the enzymes because that's really complicated. They had a general lack of parameters that they really understood the control of. And there was a lot of intellectual property involved that had several Silicon Valley companies at each other's throats. So there's a whole bunch of systems analysis stuff tied up in here, and the things that I'm going to tell you about are ways that we did it right, ways that we did it wrong, but all the way through keeping in mind that root cause failure analysis concept. So the way that DNA sequencing worked in the first generation, actually it was the second generation, but the one that we used for the Human Genome Project, was to make a whole bunch of pieces of DNA based on a person's genome, each one base longer than the last, and put a dye on the end of it that was a different color. So if the base was a G, it was black. And if it was a T, it was red. And if it was an A, it was green. And if it was a C, it was blue. And then they built a separations platform that could separate all of those components from one another and read out 1,000 bases at a time. Well, you know, the human genome is like really big. So if you're only doing 1,000 bases at a time, it takes you a long time, and it did. It took a long time. But this is the technology that we used. We made all of the different fragments using an enzyme, each one ending in a different colored base, and then we separated them all from one another, and we looked at those colors as they came by the detection window, and we got the DNA sequence out of it. Well, there's chemistry, there's optics, there's separation science, there's biology, enzymology, all kinds of different fields all at play simultaneously. And to make this system work required lots of failure. So here's how it works. You build every different length of matching the person's DNA that you're going to sequence. You put the dyes on the ends, you separate them all from one another, and you get the sequence, just like I described. And it looked like this when you get the raw data off the machine. And I can tell you that this is the M13 genome because there's a quartet at 305 bases. So if you look at these things for a long time, you get really good at it. I have a friend, his name is Erik Nilsson, he runs a biotech company up in Seattle, and one of the tasks that needed to be done was we needed to get quality metrics on these separations that we were using to prove that the instrument was running correctly before we sent it to the customer. So we used the M13 genome, which is a bacterial genome, and I needed to validate all of the places where there was a single nucleotide polymorphism, which is a place where me and Dane are different from one another. We're both boys, so we both have X chromosomes and a Y chromosome. We have almost all the same bases because our heritage is about the same, but there's spaces that were different, and those are the single nucleotide polymorphisms. I had to have somebody come up with a quality metric for these separations. So Erik has a eidetic memory. So I had him memorize the whole thing. And then I had him validate in every sequence that we made on 70 different machines the position of every single nucleotide polymorphism. So more proof that the biggest computer in the room is often the one between your ears, because making a computer do it didn't work very well. But Erik could do it really well. So what resources were needed and how long did it take to develop dye-labeled separations for Sanger sequencing and to extend that sequencing out so that we could actually do it at the human genome level? So first it needed optics. Because we're separating things on the basis of the color of those dyes, turns out they're fluorescent dyes, so they're excited, but had to make dyes. So we needed to be able to scan over long distances and keep everything in focus. And a couple of clever guys, one named Chris Alexi and another one named Bob Kane, came up with a lens that allowed us to look at many separations channels all at the same time and generate very broad data. So it made our data throughput level go up by 96-fold. So if you think about how government funding works, incremental improvements on things are funded for relatively small amounts of money. But if you can make a 100-fold increase in something, you can get the big bucks. And we did. We got a DARPA contract for $30 million, and we built flat field of view confocal scanning across many channels and increased the data throughput by 96-fold. The reason it works is that the flat field of view lens allows you to scan across many capillaries. And the capillaries are where the separations are done. So this is an array that's used in the sequencing of DNA, or it was. And that was 2 dirt roads ago. And one end goes into a plate where there's samples of the DNAs that you've manufactured with the dyes on the end. And the other end has a little plug on it where you can put in reagents and so on to fill the capillaries with separation media. And then there's a window for you to look at, and that's where the scanner comes in. 16 of these arrays were put in the machine at one time and then scanned across by this magic lens. And it wasn't unsubstantial amount of nonsense to go through to make that happen. We've got multiple lasers, we've got lots of mirrors, we've got lots of beam splitters, we've got all of these different power monitoring devices and so on, and they're all in a big box. Turned out the darn thing weighed 700 pounds when we were done because it had to be so stable for the optical scanning to work correctly. And it was monumental, but no one believed that this technology would last for long enough to make it worth reducing cost. So this instrument was sold at a loss, but the reagents to run it were sold at 96% profit. It's a pretty good profit margin, right? It's pretty high. So the next platform that followed this, a big genotyping platform that I also worked on, in 1 year, sold $42 million worth of reagents into NIH campus alone, all at 96.4% profit. So it's a giant cash cow. Designs change over time, and now a scanner that can do what the 700-pound one could do is about the size of a grapefruit and weighs about the same. So things change fast in the biotech business, and all the people that said this won't last for long enough to be relevant in just a few years were right. You got to make your money when you can, and that's exactly what we did. So was it the only way to think about solving the problem of scanning over lots and lots of capillaries and monitoring lots and lots of separations? No. There was a bunch of different ways. The one that Bob and Chris came up with is on the top, and it took many hours of scanning. The next version that Bob also came up with help from Chris Alexi, took 43 minutes, and now they're down to about 5 minutes to collect the same amount of data. So these are all order of magnitude jumps in technology, and you can imagine there's lots of problems that come along with them. The first one was, can I scan fast enough? I'm going to skip over a couple slides there. So surfaces became super important. Right in the middle of the Human Genome Project, we were trying to deliver those capillaries that were the primary disposable object to customers at every major genome center in the world. Genome Quebec, Genome Shanghai, Japan, France, Germany, United States, and everybody kept ordering more and more and more as their funding started to come through from all of the different locations that were funding sequencing projects. And we found something that was pretty terrifying. We couldn't actually produce the objects fast enough. So again, here's the capillaries. If when you build something you expect to be successful, and then you build more of it, you expect to be about as successful as you were when you were building a little bit of it. Build one house, build two houses, you probably get faster at it, but you should probably still have houses that stand up, right? So our yield is the top line, and the number of arrays that we were ordering from our supplier is the bottom line. So when we ordered more, this purple scattering of data here, our success in building them got worse and worse. It's so messed up, because you got all these customers that are just hounding you for stuff, and you're making a gigantic profit off of all of it, and you want to deliver it to them, but every time you order more, you become less successful at making them. So this is a place where the discipline of root cause failure analysis really came into play. These are super complicated objects. Number one, they're made out of glass. and it's flexible. Number 2, they're coated on the inside. Number 3, they've got a laser burnt window in them. Number 4, they need to have very high electrical potentials applied to them, and so on. And every time we tried to order more of them from our supplier, more of them failed. It turned out, anybody know the Vienna sausages? You know, the ones in the can, right? So, you know, and they're all stacked in there so nice with the little ends up. So the guys who built Vienna sausages found that their factory location was going to need to be moved, and they built themselves a new factory. They moved their gear out there. They got all up and running. They made the first batch of Vienna sausages in the new factory, and everybody was there to see the process and the project and so on. And they opened the first can, and all the sausages were kind of gray. And everybody just said, damn, what went wrong, right? And so this is a story about who you ask to help you and why they know what they know. It turned out at the Vienna sausage factory that the janitor wasn't busy all the time because he worked for 2 hours at one shift change and for 2 hours at another shift change doing cleanup. So he was the guy who was the foreman of the cleanup crew and was kind of the janitorial dude that was keeping the FDA-approved facility clean, right? And so he would pick up extra hours by helping out in manufacturing. And on the way from the packaging of the little sausages in their casings to the place where they would make their way into the cans, there was always a backup because they were actually putting all those little sausages in those little cans by hand pack. That's a really weird job. I'd like to actually have it on my CV, sausage packer. And so there was always a backup. So he would push the carts into the furnace room, and he'd pile them up along the wall of the side of the furnace room, and the furnace room was warm. And it was that aging time that brought out the pink color of the Vienna sausages, and as soon as he wasn't there to push them into that room, they weren't the same color anymore. They had to build a room to mimic what had been going on in the furnace room. And maybe they got to push it, I don't know. So exactly the same thing, same sort of thing was happening with capillaries, because there was a room, and every time a reel of capillary would come off the capillary making machine, they'd put it up on a shelf, and then the one that they had put up many weeks earlier, they'd take out, so it's a first in, first out, right? System. And every time we would order more capillary, could they instantly generate more? No, they couldn't. So they'd empty the room. So now that storage time where the capillaries were just sitting there in reels after they had already been coated and were ready to be made into arrays got shorter and shorter and shorter because we kept ordering more and more and more. So delta S is entropy, and it turns out that all the little polymers that we were sticking onto the insides of the capillaries needed to lay down. And they needed to do so in a relatively slow way, but in this case, we could mimic their aging by simply increasing the temperature. So now the room became a little 60-degree Celsius oven, and the capillaries would all go into that room and get aged for 2 hours at 60 degrees Celsius, and then moved into the storage room, and all of them worked perfectly. So short storage time, bad. Well, how much storage time do you need? It's a pretty convincing curve, man. You look at it, and if the lag time in that room is 12, 13 weeks, you're kind of maxing out the effect. And if you leave them in there for 1 week, 60% failure, 40% success in making arrays. So you're just messing yourself up by not leaving them in there long enough. So we implemented the artificial aging by increase in temperature method and solved this weird problem, except for that dip in the middle. And I'm going to talk about that in a minute. So this is where you can't be a snob at all. Like the Vienna sausages, dude, I mean, they got saved by their janitor, right? Okay, so you really can't be a snob about this. So how much did it cost to collect the metric and identify the problem? It cost $0. I asked the right question, I got the data, I looked at it, and there it was. It didn't cost us really anything, maybe an hour's worth of my time after 6 weeks of dorking around with a bunch of other stuff, right? Who had the data? Not a PhD chemist, not the glass guys, not the people who designed the coating, not the people who ran the laser that burnt the windows into them. The accountant was the guy who had the data that we needed to solve the problem. That previous slide is accounting data. Well, why? Well, in a factory that's run well, there's a station, bleep, with your barcode reader. There's an operator, bleep, with your operator badge. And there's a product, bleep. And so every time something would go into that room, it'd get recorded. And the data that helped me solve the problem was from the accountant because he cared about how long things stayed in inventory and how much time there was in our process and whether or not process improvements based on the availability of cash, as an example, would affect things. So a PhD chemist had nothing to do with it. I just happened to be the lucky recipient of some excellent data from an accountant. So lack of control. We had this November curse, and everybody knew about it at the company. By the time we got 3 years into the Human Genome Project, it had already happened twice. We got to year 3, sure enough, November, couldn't make any capillaries. Really irritating, right? Because it was obviously seasonal. I mean, people just, well, it's November curse, we might as well just stop. So let's look at that piece of data again. There it is. November comes along. Yields fall way off. We also order less because we're getting close to Christmas. Turned out that that didn't have anything to do with it. Right? That was just a false piece of information that had to be weeded out in the process of following the method. So when we built the lab, we originally built it without a clean room. And then as we got more and more sophisticated with our methods, we realized we needed a clean room. And the place that we wanted to put the clean room was where all of the deionization tanks for the deionized water for the facility were sitting. So we moved them outside, put them outside the wall, put a tube through, put a little fence around it so that it wouldn't get, stolen. Not that Silicon Valley has that much crime, but it just. And during the winter, the sun is low in the sky and there's a shadow and the water is cool. And during the summer, the sun is high in the sky and the water would always come out of the tap, the DI tap, warm. Everybody noticed it, right? But nobody really thought about it. But when did the tanks actually go into the shadow? November. So summertime, temperature high, wintertime, temperature low. When it makes the transition, there was an algae bloom. There's algae in water everywhere, right? You just can't even get it out, right? Even the city's treated water and everything, it's got algae in it. If you leave a piece of Tygon tubing on your kitchen faucet, you'll see it turns green in a couple of weeks. And despite all of the chlorination and all that stuff, there's algae in all water. So there was an algae bloom due to the change in temperature in those tanks. All those algae then died as the water got even colder. They all broke down into their little component pieces. Their little component pieces were glycoproteins and glycolipids that really like to stick onto surfaces. And the surfaces of the capillaries were being affected in a way that made them not work correctly. They had too much eddy currents in them. And so people would fill their DI water bottle, they put it down in front of their megabase, that's the instrument that we used to sequence the human genome. About 46% of it was on this instrument. And, or instruments like it, I should say, not this particular one. And they would fill up their tubes with that bottle, and frequently they'd go and refill it without emptying it again. So there was a double whammy there. Because if you don't empty it completely, then whatever's in there just got more food, right? And the breakdown products of all those things that were caused by the sun going behind the wall contaminated our product and made it so we couldn't deliver in November. Really a bummer, right? Bad success. So everybody says, yeah, you know, organisms, they grow in water. And we need water, and all the organisms on Earth need water. And so, you know, this is applicable to things where there are water-based systems. And everybody kind of puts it off as like, it's kind of obvious that that might be a problem. At the time, it didn't feel obvious. So, unfortunately, a bunch of our customers noticed that they were getting an awful lot of Pseudomonas sequences in with their human sequences. Well, Pseudomonas is a bacteria that grows in topsoil. There's about 15,000 varieties of Pseudomonas. And this particular one loves to eat acetonitrile. So in the process of making the reagents that we use to sequence DNA, primers, oligonucleotides, short pieces of DNA, we use a lot of acetonitrile. This particular bacteria loves acetonitrile. It's its food. Anything with a nitrile group on it, this bug likes to eat. And in addition, it can do its eating of your solvent even when there's no oxygen present. So it's probably digesting the glass along with it. So this bottle sits on the windowsill of my old office at Illumina down in San Diego as a reminder to people that just because you're looking for something that's a problem in your system and you think that nothing grows in it, you're probably wrong, because life loves a challenge, right? So this section's called What's That Measurement Again? So a customer came to us and they wanted to order 35,000 short pieces of DNA. And as a test to see whether or not we were good, they ordered 3,000. And 3 days later, from Shanghai, China, they sent me back 3,000 lanes of slab gel data. This involves pouring something that's about the consistency of Jell-O into a form, making little wells in the top, putting samples into them, running them through the gel with an electric current, and then getting some kind of readout from that on a scanner normally. So there must have been some army of postdocs at Genome Shanghai, all with pipette men, doing this. I mean, they did it in 3 days, 3,000 lanes. I mean, it's incredible. I can't even open, 3,000 cans of Coke in 3 days, right? And they did this huge complex process. And the way that they labeled the DNA so that they could see it was with what's called an associative dye. It's a dye that the DNA wraps around like this, and it makes it glow. It's a minor groove binder for the experts in the room. And they're great, but they don't work the way you think they're going to. So they showed up at our office, flew from Shanghai. A couple days later, after delivering the data, I offered to have them in, and to show them why I knew that what they had measured was not quite right. And so they showed me a piece of data that looked like this, that said, this is the desired product. And for some reason, these sequences in here all look like failures. It looks like I made this and this and this and this and this. This is a separation of a complex mixture. And this is the product that I wanted, and therefore your synthesis is bad. I felt really terrible about this because I had to show them that it turns out that where the G's are, so DNA is made up out of A, T, C, and G, where the G's are, how far apart they are, how many of them there are, all really dramatically affects how bright the fluorescence is when you're using an associative dye. So what they had in fact measured was not the quality of the oligonucleotides that we had given them, but instead was the quality of fluorescence generated by the sequences that they requested. There was absolutely nothing wrong with the materials. In fact, there was exactly the same amount of this as this because of the way that the dye interacts with G's in the sequence. And it turns out that the instrument function looks something like that, where the system of measuring was imparting nonsense to the information that they were trying to glean. Well, they were really impressed by that, and they bought the 35,000 oligos. This business had copious personalities going on in it. And powerful guys. Craig Venter, David Barker, all these guys that were the progenitors of the genetics revolution were all in different companies, and they were all butting heads all the time. And one of the key measurements that they wanted to be able to make was what your genotype is. So how are you different from you? And everybody was beholden to a company called Affymetrix. Affy was the big gorilla in the room. They trampled on their customers all the time because they were the only company that had invested the $145 million in building a factory to generate a platform that could measure thousands of genotypes at a time, tens of thousands. And the patent that they had was for how many DNAs you could put down in one place. And they were very sure that the fact that they had written this thing so carefully was going to save them. One of the things that they put in there was that you put them down in an indexed way. So let's say you're playing solitaire, you lay all the cards out, you put them down in an indexed way. You know where you put the card down, right? Well, if you don't know where you put the DNA down, then you didn't put it down in an indexed way. You didn't say, I'm going to take this piece of DNA and put it here, and this piece of DNA and put it here. If you just put them down randomly, then they aren't indexed. So in the quest to beat their patent, people realized that you could put the DNAs all down in random order, but attach them to little beads so that they were all distinct, and get around Affy's patent. Well, if you put them down in random order, how do you know which one you put where, right? So what we did was we sequenced them off of the beads. So we actually used DNA sequencing to find out which bead had which piece of DNA on it. And if you do that, you get some data that looks like this, where all of the beads that don't have a fluorescent molecule on them are over here, and all of the beads that do have a fluorescent molecule on them are over here. And there's a nice separation between them. What about these guys down here? Well, not as good, right? They aren't as well separated from the ones that are off. On, off. This is a lot of beads that are in this piece of data. If you look, it's 369,000 beads, right? So because we had to sequence them, because we had to measure them and put it on a CD and sell it with the product, we already knew how the product was going to perform when it was in the hands of the customer. So did I give the customer the crappy signaling beads down here? No. I cut those off. Why could I do that? Because I had to find out what was on each bead. I had to measure them before I could sell them, and AFI couldn't do that. They couldn't measure their product before they sold it. All they could do was statistical sampling. And they couldn't throw out any spots because they didn't know which ones to throw out. So in order to make this group more distinct from that group, just cut off everything from here over. Leave them out. Don't give the customer access to the bad signaling beads. So then some nice folks at one of the government agencies came along and they did a paper where they compared what are called technical replicates between the different companies. The ones on the top are AFIs. The ones in the middle are Agilent's, David's. And the ones that are on the bottom are Illumina's, my companies, right? So how does biotech get money? Well, biotech gets money originally from venture people, right? Venture capitalists, because it's too risky for banks. And they get maybe some of the startup money from government agencies like we did, but very little. In fact, there's no real serious connection other than a bunch of SBIRs that took care of little pieces of this whole puzzle along the way. But if you get money from venture capital and then it transitions out into a stock, institutional investors are really important at that point. So all your financial people in the room, if there is only one pot of money to draw from in institutional investing, then there can only be one winning company, because that institution is going to put all of their money towards the people that are better, and we were better. So the stock price in our competitor is the one on the left, and our stock price is the one on the right. So this is what happens if you write a patent and you want to be clever and you try to protect yourself. You spur on competition. And so the problem of producing the best and fastest technology that AFI was confronted with was too exclusive, and they pushed other people to do more and more and more without them. And eventually that yielded their collapse as a company. Their stock is worth about $6 right now. So, you know, what do we have to look forward to in the world of DNA analysis and so on? Well, you know, sequencing pretty soon will be used for absolutely everything. And why is it? Well, you looked at that really complicated bead data that I showed you. It's got 300,000 objects in it. It's really complicated. You have to be able to put, in order to make that measurement, you have to be able to put 64,000 objects in a tube exactly the same every single time. That's really hard to do, right? So getting to that technology was hard, and getting to sequencing turns out to be easy because all you need is reagents and a scanner, and there's a lot more profit in it because there's a lot less monkey motion along the way, and soon that'll be how we do all analysis for DNAs, will be sequencing of one variety or another. And now the platforms don't use any hardware really at all except a surface for the DNAs to self-assemble on, and then a scanner to measure them. And it's all reagents served up at, 98% profit. It's great, great business. So can you be an expert and critical thinker and work across lots of different fields? Yeah, absolutely, right? So one of the really interesting things about molecular dynamics and Illumina, the two companies that really powered a lot of the genetics revolution, was that it was the first time where analytical chemists and computer science engineers and mechanical engineers and optical engineers and molecular biologists and microbiologists all had to work together, and they all have different vocabularies. So vocabulary normalization is a huge deal when you're trying to make people talk to one another that have never done it before. So the amount of time saved by knowing each other's vocabularies, which I measured for the company by looking at the first platform and then the second platform, was 30%, 27.9% faster the second time, because everybody knew how to talk to each other. And if they said this word, everybody in the room knew what it meant, right? And on the failure side of it, all different kinds, financial, optics, surfaces, the November curse, and so on, following that root cause failure analysis method and asking yourself why things happened, and then trying to design more and more clever tests to get further and further over to the left, is only done by what Noland clearly identified as there's somebody smarter than any one of us, and it's all of us, right? And it really requires that collaborative thing. And again, that whole deal about not being a snob about where you get your data and being ready for everyone in the room, except the guy that figures out the root cause, to be wrong. It's also good, I think, more generally, to have a blame magnet. And this is a repeat of the first slide. But it's good to have a blame magnet in the room because undoubtedly some of the failures are designed into the product or into the system. And if there's a lot of finger pointing and blaming going on, it doesn't make anybody want to play. And in fact, the developers are probably some of the people that you need data from. So it's great to have somebody leading the drive towards root cause failure determination that's able to absorb a blame easily. And I do it all the time. And it's a lovely way of looking at it because it doesn't hurt my feelings at all, right? So I hope that you learned something about failure analysis, and I hope that you learned some strange facts about how screwy things can be, from Vienna sausages to complex biotech platforms. And I'll entertain anybody's questions if you have them, or we can just all have cookies.